quick review and reminder of our synapses before we break out and do an activity on how we can use drugs to target different issues with our neurons. So as a reminder, when we were talking last time about the synapse, we were looking at the fact that we have a presynaptic neuron, a postsynaptic neuron. They signal to each other. An action potential running down that presynaptic neuron releases neurotransmitters and has an effect, a potential on our postsynaptic neuron. So we talked about the fact that these are graded potentials and they can sum together to figure out whether or not our second neuron is going to release its own action potential. So when we left off, we were talking a little bit about modulation, all right? I think this is where we were. Is this a confirmation? Yeah, about here. Okay. So we were, am I wrong?
I think we're one back.
Okay, one back. Okay. So for frequency coding, so we are thinking about the fact that we're creating this potential for an action potential, an excitatory postsynaptic potential, or an inhibitory postsynaptic potential, preventing our next action potential by receiving a signal from our first cell, by receiving a signal from our presynaptic cell. So the degree of depolarization that we're going to get is going to be signaled by the frequency of the action potentials. So basically, how many signals that we're going to be receiving. Okay. So summation is going to affect depolarization, right? Like, because we can add together the effects of our postsynaptic potentials of receiving these neurotransmitters, depolarizing or inhibiting. And this is then going to affect how many action potentials we set off from the second cell as well. Okay. So if we have more action potentials coming down to our synapse, we're going to actually end up releasing more neurotransmitter, and then we're going to have greater inhibitory postsynaptic potential or excitatory postsynaptic potential in the next neuron, leading us to firing off potentially greater or fewer action potentials from that neuron as well. So basically, one thing to realize here, right? We talked about the action potential as being all or nothing, right? Positive 30 or not setting off an action potential at all. But when we think about the synapse and the axon terminal, there's not like a set amount of neurotransmitter for sure that we're going to release. There, we are going to be able to release more neurotransmitter or less neurotransmitter. So as we are creating this frequency of action potentials coming down our neuron, we're getting a certain amount of depolarization in that axon terminal, and we can change the amount of neurotransmitter that gets released. Okay. So now to the modulation. Okay. So we can regulate how our synapses are communicating with each other by having basically extra signaling neurons coming in from the side, right? So in this example, what we see, so we have our presynaptic neuron, and we have our postsynaptic neuron, right? So that's the pathway we've been talking about thus far. But we can also see here that we have a modulating neuron coming in. Okay. So this is an example of when we would see an axo-axonic synapse. So here we have a connection between the axon of our presynaptic neuron in this first pathway, and then the axon terminal of this modulating neuron, right? So we have one synapse here in this pathway, but we have another synapse here, an axo-axonic synapse, allowing us to modulate what's going on between these first two neurons in sequence. Okay, so we can do one of two things. We can either facilitate or inhibit signaling. So presynaptic facilitation, we're going to make it easier to send a signal. Presynaptic inhibition, we're going to make it harder to send a signal. So we're kind of jumping in at the end here and modulating or adjusting this pathway. Okay. So what we see happening here is kind of like when we were thinking about convergence at the end of our last lecture. right? So here we see a neuron receiving signals from D and C. So that's our convergence portion. And we can see that neuron E is modulating neuron C, right? So E here we see is not coming in and acting on this neuron X, actually connecting to that axon terminal of C, right? So when E fires, it has an effect on the amount of neurotransmitter released by neuron C. So we can see that when there's an action potential coming just down neuron C, actually, let's keep our coloring consistent. So when we have an action potential coming just down neuron C, we release a little bit of neurotransmitter. But if we also have an action potential coming down neuron E, we are increasing the amount of neurotransmitter that we're releasing. Okay? So we could look at that in terms of changes to the membrane potential on neuron X as well. So what we can see happening here, right, is that we have neuron C fire alone, not enough to get us to threshold, right? Neuron D fires alone, also not enough to get us to threshold. E fires alone, it's not even touching neuron X, so literally nothing happens. But if we have C and E fire together, we see that we do reach threshold and neuron X would give off its own action potential, right? And we know that E is only modulating C here, right? So if we fire D and E at the same time, we don't have the same additive effect because neuron C has to be firing in order to have an effect from neuron E. If E fires on its own without C, doesn't matter here. Okay, so that would be our presynaptic facilitation. So we're facilitating the release of additional neurotransmitter here. Now, in our presynaptic inhibition, right, what we see is we have neurons G and F, right, in the same place as D and C originally. And now we have neuron H modulating neuron F, right? So we're modulating neuron F the way that E was modulating neuron C in the previous diagram, okay? So in this example, F releases a certain amount of neurotransmitter if it fires alone, right? But if it fires at the same time as neuron H, right, neuron H is essentially going to suppress that release of neurotransmitter, right? So we're suppressing that release of neurotransmitter from neuron F. So we have less neurotransmitter released. So this is going to be our presynaptic inhibition. So presynaptic, because we are talking about these presynaptic neurons, right? These ones that I've circled are presynaptic. Here in yellow is our postsynaptic, right? So presynaptic facilitation or inhibition is affecting how much neurotransmitter that presynaptic neuron releases, okay? So we can see on our membrane potential, neuron F fires alone, little bit of depolarization. Neuron G fires alone, little bit of depolarization. H, nothing at all. F and H, despite the fact that F did depolarize us a little bit, has no effect whatsoever because H has suppressed it, it's inhibited it. Whereas F and G together without H would get us up to that threshold, fire off an action potential. So we're modulating or changing the effect, adjusting the effect with these modulator neurons. So when we think about our different types of synapses, the axo-axonic connections are going to be selective, right? They're going to be really exciting or inhibiting that one synapse, that modulation that we just looked at. Our axodendritic and axosomatic connections are going to excite or inhibit the postsynaptic neuron, but they're not going to be as selective here, right? And so like part of what we can think about here, I'm going to draw us a neuron. Okay, So thus far, we've been drawing our neurons like that, like cell body, stoma, axon terminal, just one. But to understand the selective versus non-selective, we're going to note that our neurons actually have multiple axon terminals, right? So if we have an axo-axonic connection, right, we're thinking about another neuron modulating just this one synapse, right? So it's selecting just this connection here, right? This axo-axonic connection is exciting or inhibiting that one synapse. Whereas if we think about a neuron over here, right, signaling to the cell body, so this would be an axosomatic connection. If we came into a dendrite, the dendrites are just those processes sticking out. It would be the same kind of situation. This would excite or inhibit that whole neuron, right? So it's affecting the whole neuron because it's affecting whether we're setting off an action potential to all of these terminals or not. Whereas this axo-axonic connection is just affecting this one synapse. So that's what that's talking about with the selective versus non-selective. So if we summarize what's going on here in our whole sort of picture, we have our presynaptic neuron, right, firing off an action potential in response to a signal, right? That action potential comes down to our axon terminal. It's going to open voltage-gated calcium channels, and we can see this happening at the other end of this neuron as well, releasing our neurotransmitter. Okay. So if we release an excitatory neurotransmitter that excites the next cell, depolarizes it, makes it more likely to set off its own action potential, right? So we can think about an excitatory neurotransmitter as one that would, for example, open up more sodium channels, whether directly or indirectly. Okay. So we're going to open these chemically gated channels, or potentially with a G protein if we're talking about our metabotropic receptors. And in that post-synaptic cell, once we've opened those channels, we're creating a change in the membrane potential because we're talking about an excitatory neurotransmitter. We might think about sodium channels causing depolarization, which is an EPSP. Okay, so this EPSP is a type of graded potential. So we're actually trying to figure out if the axon hillock of our postsynaptic neuron is going to be affected. So we'd have to sum together those decremental, right, decreasing changes in membrane potential that we may remember from our discussion of graded potentials. We're summing them up at the axon hillock, determining whether we set off an action potential or not. Right, so this first segment is what we have to depolarize to threshold, this axon hillock. If we do polarize it to threshold, in that case, we are going to first open our voltage-gated sodium channels by opening up those activation gates. We're going to see our depolarization spike at positive 30 as sodium is rushing into the cell. We have a delayed response from both the inactivation gate of the sodium channels as well as those potassium channels. So as we peak to positive 30, the inactivation gate of the sodium channels is swinging closed, stopping us from spiking higher. The potassium channels have opened, allowing potassium to start to rush out of the cell, repolarizing us and returning us to the resting membrane potential eventually. So that's kind of our whole view of how our neurotransmitters and our synapses and these connections work. So we want to remember our spatial summation, our temporal summation. We can think of that axon hillock as an integrator because this is where this summation is happening. So we are not going to move on to our central nervous system today. Instead, we're going to do a little sleuthing into the effects of psychiatric medications and mental health stuff. So I'm going to drag and drop this into our folder. And I'm not going to be asking you to like memorize anything that we're talking about today. This is to practice our synapses and our understanding of these connections. Extensive file structures. So we're partially talking about this because this is a topic near and dear to my heart, but also because this is a real problem that affects a lot of people these days. So before we get into this, I have also linked into this document, the PSF Mental Health and Wellness Resources. If any of this feels extra personal to you today, go talk to someone. It can be really helpful. We have a lot of people who are able to help, and it has some like cool stuff that you might not expect on here. So like a very long document, but sign in again. All right, so we have a long mental health resource list here. We have a bunch of apps that you can use. We have resources for specific communities. Oh, for this? No, you have the slide here. I just dropped them in there. So you can access all of these resources through the school website. They're all around here. You can refer friends, all sorts of good stuff. Okay, so this is here. All right, so in 2020, right, like this got extra bad, our mental health crisis in this country. In the age group for most of you, 18 to 25, 1 in 3 people reported experiencing a mental illness in 2020. 1 in 5 people reported that was due to the pandemic. 3.8 million people were struggling with thoughts of suicide. So this is a very, very real issue. And medications are something that can help depending on the person, depending on the condition. In general, with mental health, there's like 11 years between the time that people have symptoms, aka right, the time people start feeling really bad, start having problems, and the time they actually get treatment, which is a big problem. That's more than a decade of potentially feeling awful, right? Prescription mental health medication use has gone up in 20% in the last year. So presumably this means that lots more people realize they need help. But that is also why we're going to talk about how some of these things work today. Okay. So when we were thinking about neurotransmitters, we mentioned 2 of them in our actual slides for our coursework. We talked about glutamate and GABA very briefly, right? So glutamate and GABA are our major excitatory and inhibitory neurotransmitters in the brain. So these do have things that they're associated with, right? So glutamate is often associated with learning. GABA, inhibitory neurotransmitters, are often associated with anxiety disorders, things like epilepsy. But we have a lot of other neurotransmitters in the brain that have complicated effects on your brain, on signaling, and on these various pathways. So this is a table of some of these neurotransmitters, their abbreviations, and some of the behaviors that are associated with problems or diseases related to each of these neurotransmitters. Okay, so just a reminder of our way our synapse is working, because many of the drugs we're going to be talking about affect synaptic communication, right? So we have our action potential coming down the synapse. That depolarization triggers our voltage-gated calcium channel. Calcium rushes into the cell. Our vesicles with our neurotransmitters inside them then dock to the membrane, releasing our neurotransmitter. which binds to a receptor, potentially an ionotropic receptor, potentially a metabotropic receptor. Our receptors actually each have names. They're specific to specific neurotransmitters. They have specific mechanisms of action. We have a cellular response, ultimately going to cause either a depolarization, excitatory, or a inhibitory effect, so like a hyperpolarization. That neurotransmitter should then be broken down and cleared or taken back into our original cell. And various drugs are going to potentially affect these pathways. So they may be affecting reuptake. They may be affecting these receptors in our postsynaptic cell. Some of them actually target the presynaptic cell as well, targeting various types of voltage-gated channels. We can basically try and modulate our neurons ourselves by targeting these various pieces of the system. Okay. So we're going to first talk about SSRIs, or selective serotonin reuptake inhibitors. So these are super common. So we have some examples at the bottom here. All right, so the way that these are affecting our synapse, right? So in our presynaptic cell, we have our neurotransmitter stored in the synapse. Right, so we have our action potential coming down. Our neurotransmitter should be released as the calcium receptor, voltage-gated calcium channels are triggered. We have neurotransmitters released, and the receptor for serotonin here is our 5-HT receptor. Okay. Now, what the selective serotonin reuptake inhibitors do is they block the reuptake of this molecule, right? They block the reuptake of the serotonin, which means that we're going to have essentially more serotonin in our brain. We're going to have more serotonin hanging out for longer in the synaptic cleft, which means that we can have more of an effect here, right? So if you have really low levels of serotonin, or you're having trouble producing serotonin, you're having trouble releasing serotonin, when we block the reuptake inhibitor, this can normalize the amount of serotonin by allowing that serotonin that you are making to hang out here longer, allowing those receptors to get more serotonin. And that's a brief explanation. We're going to dive in deeper in our sort of activities potentially. Okay. So here we have a couple of drugs listed along with their mechanisms of action. So we're going to use some of these tables together, as well as do a little bit of our own sleuthing to each sort of pick one drug and dive into how this affects our neurons, right? So the way we read this one, right, is let's pick Adderall, right? So here we see that Adderall is increasing the release of dopamine and norepinephrine used for ADHD. Also an agonist, right? Agonist means it's like increasing. Antagonist means it's like decreasing, essentially, for our purposes. So increasing those effects of the dopamine and the norepinephrine. Right? So as we do these activities, you're going to trace through what effects this is going to have on the cell, where this is targeting, what a synapse without this looks like, what a synapse with this looks like. So we have a bunch of tables we're going to use, and these are also located